Hello everyone, welcome to Emsa. Today in this video we will be discussing about one of the technique, that is Emsa. It is also known as electrophoretic mobility shift assay. So by using this technique we can study the interaction between the protein and the DNA. So before that we have to be very clear about the gel electrophoresis. What we do in gel electrophoresis is that we separate the DNA, RNA as well as protein according to their molecular weight as well as molecular size. So if the molecular weight is more, if it is high molecular weight, if the molecular weight is more, so the DNA cannot pass through this well easily. That means we can get the band near the well. But if the molecular weight is low, we can the DNA or the protein, it can migrate through this gel easily, and we can get the band in the end. That means away from the well. In the similar way, if the molecular size is high or it is large, then it cannot move through this gel easily. So we can get the band near the well. And if the molecular size is low or it is small, okay, during that case we can get a band in the bottom or away from the gel. In this way, the Emsa can be performed for the detection of the binding of the protein and the DNA. So let us see what happens in case of a DNA. There are various types of proteins which are interacting with it. Let's take during the process of DNA replication, the DNA is interacting with various proteins, like it can be a DNA polymerase, it can be a DNA polymerase, it can be some of the helicase, it can be a single stranded binding protein, it can be gyrase. These are some of the proteins which are interacting with the DNA for performing the replication. In the same way, there is a protein called histones which are interacting with the DNA for providing the compactness. And there are various transcription factors which regulate the DNA by interacting with it. So let us see how this Emsa has been done. So when we want to study that whether a protein is interacting with the DNA or not, in that case we are performing this kind of technique. So for that, what we have to do, first thing we need a DNA. This DNA need to be radio labeled in the 5' end. In the 5' end it is radio labeled or it is fluorescently labeled. Why it is done so? Because whenever the DNA is passing to the gel, we can easily identify that this band is present in the gel. This is the reason why the radio labeling is done. So in one well of the gel, what we are adding is we are adding the labeled DNA. This is the only DNA. As only DNA is present, it is having low molecular weight. Due to its low molecular weight, it can easily migrate through this DNA, and the band which we will get is in the bottom, as it is easily moving. The next thing what we have to do is, suppose this is a protein A and we want to study that this protein A can bind with the DNA or not. So in that case, we have to take a tube and mix the protein A and the DNA. If this protein A and the DNA are interacting, that means they are increasing its molecular weight, and when we do a gel electrophoresis, we have to load the sample in the well. If this protein and DNA are interacting with each other, they will increase its molecular weight, and when the molecular weight is increasing, it cannot pass through the well easily. It will the passing will be slower. So here we are getting the band near the well. So when we are keeping the DNA, only the DNA when we are adding in the well, we are getting the band here, that is in the lower region, but when there is a interaction of the protein, there we are getting in the upper portion of the band, of this band. So there is a change of this position of the band. That is the reason this Emsa is also known as band shift assay. Let us be very clear with some other examples. Let us see here, we are having some radio labeled fragment, and this radio labeled DNA fragments are run in a gel electrophoresis, in which we get single band which is present in the lower portion or towards the base or away from the well. In the second condition, what we have is there are various types of proteins. The proteins are of different size or of different molecular weight. They have been added to the sample of the DNA. So Here we can see that the C1 is interacting with this C1 protein which is present. It is interacting with this DNA. The C4 is also interacting. C2, C5, C3, C6 proteins are interacting with this DNA, but they are having different molecular weight. So the protein which is having low molecular weight, it will move faster. Here we can see that the C6 which was present, it was having the low molecular weight. The C6 is a protein or cell extract in which the protein is present. It is having the low molecular weight. That is the reason it can migrate easily. So here we can see that first of all, that's the free DNA will come, because whenever we are adding some of the proteins into the DNA, there is no chance that in every DNA everything will bind. There will be some of the free DNA present in the sample. So the free DNA will be present, they will be present in the end. The band which will be present, they will be in the end. The next comes is the C6. Why here the C6 is present? We can see here that the C6 is having the small protein of all these proteins. So here the C6 came here, and according to the size they are being arranged. Here we can see that the C1 is having high molecular weight, or the size of the protein is more. So here the C1 protein or the band of this DNA is coming here, because the C1 protein is interacting with it, due to which it is having the high molecular weight. In this way, C4 is having small size as compared to C1, and C2 is smaller than C4, and C5 is smaller than C2. In this way, the C1, C4, C2, C5 and C3 has been arranged. The band of this DNA has been arranged. So we have to put the sample. The first step is that we have to put the sample of the single DNA as well as the DNA in which the proteins have been added. The next thing, we have to do the gel electrophoresis. After we run the gel, we have to take this and see it in a autoradiogram if it is radio labeled. After that, we can see if there is change in the position of this DNA, then we are confirmed that the DNA and protein are interacting with each other. So let us see another example how this can be done. So here in this well, we are having in the 3 well, here the 3 well is used, and in the 3 well there we have provided the DNA. So here we can see that in the lower portion, as the only DNA is present, this DNA is having low molecular weight, and this is giving the band. In the first well, there is no proteins, like there is no protein A, no protein B, so there is no band present here. In the second well, here we have added only protein B and no protein A is added here. So due to the presence of protein B or the interaction with the protein B with the DNA, it is showing one band. It is shifting its position due to high molecular weight as compared to the single DNA. In the third well, we can see that the protein A is provided but protein B is not provided, and here we cannot see any of the band shifting, because this protein A which we have provided, it is not interacting with the DNA. So from here we can conclude that only protein B is interacting with the DNA and protein A is not interacting with the DNA. In the similar way, let us see other example. In this we can see that in every well there is DNA present. So here we are getting the band. These are the 4 bands of the DNA, only the free DNA. In the first well, we don't have any proteins, like the protein A is not present as well as protein B is also not present. So there is no shift of this band. In the second well, we have added protein A and no protein B is present. And because this protein A is interacting with this DNA, it is showing the band which is upper to this. That means there is a shifting of this band. When the protein A is binding with this DNA, it is increasing its molecular weight, due to which there is a shift of this band. In the third well, there is no protein A, no protein A is present, only protein B along with DNA is present. And we can see this, there is no interaction of this protein with the DNA. In the fourth well, we can see that there is a protein A as well as protein B and DNA. In this case, we can see 3 bands. So from this well, we can confirm that this is in this size we are having protein A. So here also we can conclude that this is a band for our protein A. and this band is having higher molecular weight as compared to this single protein A. So from here, as we have given two proteins, protein A and protein B, from here we can conclude that this is the band for protein B. So what happens in this case? When protein A is given, it is binding with this DNA. When protein B alone is given, it cannot bind with the DNA. When protein A along with protein B is given, it can bind with this DNA. That means whenever the DNA is present, the protein A can bind, protein B can bind, the protein B cannot bind, but when protein A along with protein B is given, it can bind. So how can it be possible? So this protein B which is present, it may be a transcription factor and it requires some of the proteins for the interaction with this DNA, because it is not having the DNA binding domain. The DNA binding domain is not present with protein B. It is just a regulatory factor. That means it requires a protein A for its interaction, or it will bind with the protein A and the protein A will bind with the DNA and it will regulate the work of this DNA. So when these two proteins are interacting with a single DNA, it will be having high molecular weight as compared to the DNA which is interacting with only the protein A. This is how we can see, or we can find out that whether the proteins are interacting with this DNA or not. This is all about the Emsa technique. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.